We're going to be talking about different types of contrast agents, and I'm going to lead off and give an hour lecture on scattering, absorbing, and modulating probes. And then we've got Professor Alex Way from Purdue, who's going to be talking about some of his nanoparticle contrast agents. And then finally, Yi Lu, who's a professor here at Illinois, is going to be talking about some of his probes and targeting DNA and other moieties. So hopefully it's a good chance to kind of see this perspective of biophotonics. Any questions that people have kind of before I get started? Any problems that have come up? Anything that you need to bring to my attention? Okay. So I know people are starting to feel a little bit run down after a week so of intense biophotonics, but the end is in sight. This week probably isn't going to be any lighter, but the end is near, so hopefully we'll wrap up on a positive note with our conference, a banquet Thursday night and conference on Friday. Okay, so I wanted to talk about this type of class of molecular probes that are a little bit different from the fluorescent reporters, many of the quantum dots, the types of agents that you're probably more familiar with. And this kind of segments off of some of what I presented last week about OCT. So in developing OCT, we had to look for other types of contrast agents that might be useful specifically for OCT. So, questions at the end, or can we ask questions? Oh yeah, of course, sure. Yeah, feel free, just stop me. I tend to talk fast, but put up your hand or shout out. Last week, I covered sort of these topics about OCT, some of the backgrounds, and this lecture today is more about, again, trying to use OCT for molecular type imaging and designing contrast for OCT. So if we think about contrast in imaging modalities, we look and see that every modality has some sort of contrast agent. And these are used really to just improve the imaging capabilities, the targeting, the site-specific imaging or labeling of molecules in tissue. If we look at MRI, of course, they have gadolinium and iron oxide particles that really will change the T1, T2 contrast. In nuclear medicine, of course, there's all kinds of radioisotopes. In CT, there's ionated compounds. In ultrasound, there are microbubbles. And in optics, we have got a lot of contrast from bioluminescence or fluorescence or Raman. But what I want to emphasize today is more on scattering or absorption contrast. So this is a bit of a metaphorical diagram of a contrast agent, and it has many layers. If we look at the core, there's different sort of bases that we can build contrast agents on. And this is optical imaging in general. So there are liposomes, there are plasmon resonant structures, protein microspheres, nanoparticles or dyes. And the point of this figure is to emphasize that there's a lot more of optical contrast than just fluorescence that we're used to. So we can look at fluorescence, but there's also bioluminescence, absorption, optical activity, birefringence, nonlinear processes, scattering. All of these optical properties could function as a means of generating contrast. Now those properties can either be static, can alter that property in a permanent way, or dynamic, meaning we can change them in some way. And then we can measure with our instruments the amplitude, the phase, the wavelength dependent, the polarization dependent, nonlinear properties of all these different agents, and finally apply them to many different applications across biology and medicine. So if we look at it this way, there's a lot more to be discovered and to be thought about for contrast agents. Now, I talked a little bit last week about OCT for breast cancer imaging, and this has been a focus for us in my group. There's limitations, of course. All of this imaging has been just really structural imaging, looking at the scattering based on the changes in optical index of refraction. And this is in comparison to histology. Now, histology has the benefit of being able to, in fact, do some form of molecular imaging. They add different dyes or stains that will target different molecular structures, chemical structures, and that is a basis by which we can stain and identify tissue for diagnosis. Well, we don't have that luxury necessarily in OCT, but our goal is really to maybe have some type of contrast agent that can give us that molecular specificity of these different tissue types. So if we think about the contrast methods in OCT, there's many that we could generate. There's in fact functional or physiological types of contrast, Doppler, blood flow, birefringence using PSOCT, and then there's different ways that we can add or identify endogenous contrast. So there may be spectroscopic changes between melanin, hemoglobin. There may be ways of, because second harmonic generation is a coherent process, we can use SHG, OCT, to look at collagen or ultrastructure. And there are nonlinear techniques that look at the vibrational states of molecular bonds. And this is using CARS, and CARS, in fact, is also a coherent process that we can detect with an OCT system. So there's functional, there's endogenous. But what we have to recognize is that, OCT does not sense fluorescence. Obviously, it's a coherent imaging process. Fluorescence is coherent only over an extremely short distance, and we have to think about how we're going to design our probes, as well as design a way to detect those probes. And in fact, we have to, when we think about this, we have to be able to generate probes that are small enough so that they're injected into the vasculature, they will circulate, they can extravasate, but yet as the size gets smaller, the scattering properties of those particles become more Rayleigh-like scattering. And so we have, or more isotropic. And so we have to kind of weigh the size issues. We have to be able to specifically target these particles. Of course, they have to be biocompatible, and we only hope that that minimal detectable concentration of agents is going to be less than the toxic concentration into the tissue. So if we think about how we detect those, of course, we've got to somehow be able to discriminate them against the background and also make sure that our system is sensitive enough to detect those. So it's really a synergistic approach that we have to think about new agents and new design systems to be able to come up with these new techniques. So if we look at what's been done with exogenous probes, those are agents that are added to a living tissue to provide that contrast. We can see that they're based on scattering, such as microspheres or nanoshells, absorption, so there may be specific plasmon-resonant structures that have narrow absorption spectra. They maybe have spectroscopic absorption. They may be transient, or another technique that we've developed here called magnetomotive, where we, with a magnetic field, will modulate particles. So we're going to look at a couple of these in the lecture this morning. So all of these sort of collectively are grouped under this idea of molecular OCT, where we can now develop either contrast agents, exogenous agents, use spectroscopic techniques, or use this endogenous molecular imaging technique called NIVI to be able to sense the molecular composition of tissue. And that has inherent value in trying to make a diagnosis. Now, let's look at the different types of classes again. With OCT, there's, among scattering agents, things that might seem to work would be microspheres, micron-sized particles, liposomes, things that clearly will provide a strong scattering signal. There may be magnetic agents, such as ferrofluids that have iron oxide in them. They could be nanoparticles, or maybe we can put these nanoparticles into microspheres, which are shown here. Under these spectroscopic agents, there may be near-IR dyes. So dyes that we're not interested in the fluorescence, but we're interested in how they absorb the light in the near-infrared, and they'll change then the OCT spectrum that's recovered. And finally, these plasmon-resonant agents that will provide strong scattering or absorption spectra, such as gold nanorods. And if we're thinking about these, there actually are many other types of analogs that are already FDA approved in the US. So protein microspheres are very similar to agents used in ultrasound. A lot of the iron oxide nanoparticles are FDA approved to use in MRI. Near-IR dyes that are FDA approved, fluorescein or many other types of dyes that we use, for instance, in the eye or other cancer applications. So it always makes sense, because we're somewhat limited. We know that new agents take many, many years to get FDA approved. If we can leverage a lot of what's already been approved, that can speed up this process. So I wanted to focus on this technique for the most part of my lecture on this Magnetomotive technique. And the basic idea here is to consider that we can start with magnetic nanoparticles that are iron oxide, very small, nanometer size, tens of nanometers. And we apply some sort of alternating magnetic field externally. That's going to cause the signals, the OCT signals, to modulate as well, because these particles, as you'll see, will actually move within the tissue and change the optical scattering properties transiently. Then we go from just generating a structural OCT image to one that it actually has contrast based on this magnetomotion. And you'll see that we can not only use this for contrast, but also for making mechanical measurements and therapy as well. So here's the idea. We want to use magnetically susceptible agents because human tissue is not highly ferromagnetic. Any type of displacement or rotation of these particles is going to be induced by this magnetic field. And if this particle moves outside of the beam, or if there's axial displacement, or maybe there's rotation of an anisotropic particle, these are all going to result in modulations of the amplitude and phase of our interferograms collected with OCT. So this is just an M-mode image. So this is depth versus time. And we can see as we modulate this B field, we see that there's this modulation of scattering coming back from particles in an agarose gel. So we can indeed switch this on and off. And there's always, in much of what you do in biophotonics, if you have a way of controlling your signal by switching it on and off, triggering it, or have some sort of triggering event, that's always going to be better for you to exert control, to enhance contrast, to find your signal. So this is just one example of that. So if we're thinking about these magnetic nanoparticles, we have to think about, well, what is their near-IR scattering or absorption properties? Can they be discriminated from the background? Are they biocompatible? What's their size and molecular specificity? So these types of magnetic agents address many of these requirements quite well. So we can control them externally. They have a high magnetic susceptibility. They also overlap with the MRI, contrast agent technology. And so the susceptibility of iron oxide is quite high compared to that of tissue. So we have this potential for a lot of contrast. Now, over the years, we've been doing this for many years now, but we've looked at many different commercial type vendors for these magnetic particles. Some work, some don't. We don't always understand why, because they have proprietary ways of making these and coating these. But some that we did find that would work are for Motion Nanotech and Sigma Aldrich. You can see that they're typically, what's the scale bar here, 50 nanometers. So roughly about 20 nanometers or so in size. How this works, we take a standard magnetomotive or a standard OCT system, and what we've done is we've simply added a power supply and electromagnet in the sample arm, and we image through the bore of that magnet. And this is a really small, weak magnet that's going to exert a magnetic force or gradient on these nanoparticles. And that force is going to cause that particle to move, and as I mentioned, is going to change that interferogram. The force on that particle is really proportional to the particle volume, also to the magnetic field gradient. And so we obviously want a larger particle to give us a stronger force. But the larger the particle, the less likely it is to travel, to extravasate out of the bloodstream to get to different tissues where we might like it to go. So we're always weighing those options. But some general calculations show that with a very weak field, 0.09 teslas, this is going to exert a very weak force, 0.2 femtonewtons, on a 25 nanometer particle. And this is enough to really cause it to modulate depending on the environment that the particle is located in. So just to give you kind of a frame of reference, most magnetic fields, the static fields in MRI systems are 1.5 or 3.0 Tesla. So just really many orders of magnitude larger than what we exert for this technique. Okay, so these particles that are moving now, they're not the ones that are directly responsible for that changing in the scattering. What happens is the particle is going to be perturbing the local environment. So in the off state, we have our particle and we have our tissue. If we turn it on, that particle is going to move. It's going to change that local tissue structure. And that's what's going to change the scattering properties. So it's going to cause, again, kind of to modify the amplitude and phase of our interferogram. So this is just an overall schematic of how we integrate this. What I wanted to show or emphasize here is that this is just an agarose phantom that has these magnetic particles in there. And as we take M-mode images for different magnetic field waveforms, we can see similar changes in the scattering. So if we have a relatively sinusoidal waveform, we see this type of sinusoidal scattering change. If we have a pulse, turn on the field and shut off the field, we see these transients that happen at each transient of the field. You'll also see that, maybe recognize that there's some ringing here, and we'll make use of that for elastography applications. And finally, we can chirp a sinusoid, and we can see a similar chirp type waveform in the scattering signal. So we can exert this type of control externally on these particles in the tissue. Now, these particles aren't moving very far, so this is, again, a similar example of where we're switching these on and off. If we look at the axial displacements, it's only on the order of a few hundred nanometers. Of course, it depends on the field strength and how long we turn that on. But these are really small displacements, and we can make use of the phase sensitivity of OCT to be able to measure displacements on the hundreds of nanometers, this type of scale. So this was one of the first papers we published that where we took these magnetic nanoparticles. These were microparticles, I believe, when we started. We fed them to macrophages in a dish. Macrophages will eat anything that you give them if it doesn't kill them. And then we dispersed those macrophages in a 3D gel, and we could clearly see the magnetomotive signal coming from macrophages that had iron oxide taken up. And roughly about 50% of those macrophages that are scattered around here showed the signal, and none of them in the control signal. So this was really promising in terms of being able to identify where these particles were. This is another example where we took in vitro skin and injected particles. If we look at the structural OCT of the no agent and the tissue that had the agents, we see maybe there's a little bit more scattering that you can see overall in the structural image, but clearly not dramatic and not something that you could use diagnostically. If we took and apply our magnetomotive technique, we see that clearly those particles all light up. There's a little bit of background noise in the non-magnetic case. Is it from blood? Is it from just movement in the sample? But clearly there's a strong signal from the magnetic nanoparticles. And of course we can overlay these and we can look to see where these particles have gone or diffused through this tissue. And just another example, this was in vivo where we injected particles into a tumor, and we can see clearly collections. We can even kind of see where the needle track was as well in this particular tumor. Now, we wanted to understand better about the dynamics of where these particles go in tissue. And so we did a couple of different studies. One was, let me see where to start. So one of the questions that we had was, how are these particles really moving in the tissue relative to their environment? And we hypothesized that because the particles are moving the tissue around, then they have to be bound in some way. So the tissue, we thought, was actually acting as a restoring force. So the magnetic field would pull on a particle, would let it go, let's say, and the tissue would cause the particle to return to back to its resting position. So if that were the case, then if the particles were in just a liquid with very low viscosity, we wouldn't see any signal. So we drew this curve. We hypothesized at first that that when we had a sample that had very low viscosity, then we wouldn't see any signal. So what we're plotting here is the normalized magnetomotive signal over time from a gel that's going to be hardening. So we added, it starts off as a liquid. We added the different components, and it's going to start to harden over time. So we hypothesized we would see no signal. As that gel becomes sticky and viscoelastic, that signal would go up. And as that gel becomes completely solidified, our signal should go to zero as well, right? Because there's going to be no movement. Well, that's essentially what our experimental data showed. That at liquid state, we saw a very low signal. It kind of peaked in this viscoelastic state. And it never quite went to zero because, again, we're looking at nanometer displacements. And maybe there are, we hypothesized, even though the bulk material is rock hard, there are little micro cavities of perhaps gel that hadn't solidified, and we're still allowing these particles to move. But we started to better understand how these particles are behaving in tissue. Next, what we did was we essentially took different types of tissue from a rat model, and we marinated these in nanoparticle solutions over time. And we tracked to see what signal was coming back from these particles, from these tissues, at different time points. Now, we expected to see, as the particles would diffuse into the tissue, we expected to see kind of this front, this wave front, where the particles were traveling deeper and deeper, diffusing through the tissue. But that's not what we saw. If you look here, this is muscle and lung. At 0 minutes, there were no signal. 25, we all of a sudden see signal pretty much throughout. And at 40 and 60, it just gets brighter and brighter. And if we thought, the more we thought, the more we realized, well, if these particles are diffusing, they're not bound. So they're not going to have that restoring force. They're a freely diffusing particle is not going to show a signal. So what we hypothesized is that these particles diffuse through the tissue, and whether the concentration got slow enough or they got bound in some way to proteins or within the tissue, all of a sudden we started seeing the signal everywhere because those particles have stopped diffusing and were bound. And so then we looked at different types of tissues: lung, muscle, liver, skin. It's kind of hard to see, but liver here, the signal shot up very quickly. This is skin. Skin was low, flat, because they never diffused into the skin through the stratum corneum. But interestingly enough, the time that it took to penetrate into the tissue and the resonant frequency that we could measure with these particles all correlated with the stiffness of the tissue. So the stiffer the tissue, you could think is harder, denser. It took longer for the particles to get in, and the resonant frequency was higher. So if we took the resonant frequency, we can determine the resonant frequency by simply sweeping our magnetic field that we deliver to the sample. And we're going to see, like with many resonant frequency characteristics, when you hit that resonant frequency, you see a very strong signal. And we were able to measure that. So this was really interesting that we could start to measure mechanical properties of the tissue. I'll come back to that in a new technique. We also then started working on the problem of trying to conjugate antibodies to these nanoparticles, because we want to target these. And what we worked out was that we ultimately want to do Fc-directed conjugation. Now, if we talk about antibodies, they're usually represented as kind of this Y schematic. And the Fc portion is sort of the stem. The Fab portion is sort of the Y part, the outer part. And the Fab portion is where a lot of the active sites are for binding. Okay? And what we realized is that it was very important to be able to do this Fc-directed conjugation, because macrophages that are everywhere in the body, often in the liver, have a lot of receptors for this Fc portion. And if those Fc portions are exposed, macrophages are going to opsonize or take them up much more readily. So by doing Fc-directed conjugation, we can minimize what we believe. We minimize the uptake by macrophages. And this is going to have a significant impact when we target these. Also, we essentially will optimize or expose more of these active sites around the nanoparticle. And that will allow us to be able to target these more specifically. So we did a lot of different studies that confirm this in cell culture. And we went about showing this now in a rat mammary tumor model. And let me walk you through all this data. So we took these magnetic nanoparticles, we did FC directed conjugation, we injected them IV, let them circulate through control animals, animals that had tumors. Let me back up. So all animals had tumors. Some animals were given just control saline injections, some were given non-targeted agents, and some were given targeted agents. And what we found is that all the targeted agents clearly targeted the tumors of interest, and the non-targeted and the control tumors showed no magnetomotive signal. This was all confirmed on histology and immunohistochemistry. But if you look at this, these plots, this sort of tells it best. In vivo, we saw that there was a very significant amount of targeting to the tumor. The non-targeted saline were low. That was simply repeated ex vivo when we took the tumor out. And then if we looked at the biodistribution, we saw some interesting results. So in the targeted case, most of the tumor showed a very high percentage of the targeted agents. The non-targeted agents showed very little uptake in the tumor. The non-targeted were all collecting in the liver and the spleen, okay, as we might expect. The spleen actually had a false positive peak, and that's because a lot of the ferritin that's present in the spleen also shows a magnetomotive signal, interestingly enough. So that, even in the targeted case, was really a false positive, because that's just in the endogenous ferromagnetism of the tissue. But what this showed is that with this method, we could get a lot of our magnetic nanoparticles targeted to the tumors, very low uptake, and in the non-targeted case, of course, they were just all cleared from the liver and spleen. Okay, so that was really a positive result for us to be able to demonstrate that. These particles also could be used in MRI, and so in MRI, iron oxide particles will produce a negative T2 contrast. And this just shows that as we increase the concentration, the dose in parts per million, we see that the signal gets darker and then goes away. That's the negative contrast that we're inducing. And in fact, in that same study with the rats, we used MRI to make sure that the particles got to the tumor. And so these are MRI images of the rat. The arrows kind of point to the tumors that are present in the mammary tissue. And what is shown here is for targeted, non-targeted, and saline. In the targeted case, what we did is look at the volume change within the tumor. And after the injection, we go from the blue to the red curve. So there's a negative T2 shift. Okay, and that's what we expected to see. In the non-targeted case, it was pretty much equivocal. There was no major shift. And in the saline case, it was pretty much the case as well. In fact, it was an opposite shift, that after the particles, there was a positive T2 shift, or after the saline. So this just verifies that with MRI, we could detect that there was a negative T2 shift due to the presence of these nanoparticles. So clinically, how we imagine this working is that these particles may be used to identify tumors in the whole body, and then use magnetomotive techniques, say in the operating room, where you're looking for tumor cells and you're looking to identify the tumor with very high sensitivity and be able to resect that tumor. Okay, any questions so far on this magnetomotive technique? All right, well, I mentioned that there's this inherent link to how these particles are really bound in the tissue. And so this developed our idea of magnetomotive optical coherence elastography. Can we actually measure the mechanical properties of the tissue? And so we set about testing this idea, and the first thing we had to realize is that we had to generate a phantom that not only had the optical properties of tissue, but now had the mechanical properties of tissue. And that was really done by mixing a silicon matrix with various types of gels to tailor this elasticity. So this PDMS with this RTV as a curing agent. And we were able to get the mechanical properties of this gel very similar to tissue as well. And we then doped it with our iron oxide nanoparticles, and we also had titanium dioxide microparticles to act as added scattering agents. Okay, so then we took our OCT, magnetomotive OCT, along a single depth scan, and we looked in M mode, and we could see then as we were turning our field on and off, we could see these very small transients and these almost sinusoidal type transients when we were switching the field on and off. Well, if we look more closely at these transients, we could see that with both the amplitude and phase, we saw these transients, and we saw that they had characteristic frequencies, this oscillation frequency, and sort of a damping factor. So how quickly did that signal die off? You can see up here. If we just switch, go from off to on, we see this transient, this oscillation, and this kind of dies off. We can then fit these curves and determine the damping coefficient, which tells us the viscosity, and the oscillation frequency, which tells us the elasticity of this tissue. So we're able then to be able to sample, use these as sort of optical rheology agents to be able to characterize the mechanical properties of the tissue or microenvironment. So this was a study that was done using different gels at different stiffness. And we can see that as the stiffness of the gel got higher, going from green to red to blue, that frequency, oscillation frequency, got higher and higher as well. And this actually fit quite linearly to this plot of relaxation frequency versus the square root of the elastic modulus. So somewhat very predictable then. Now, when we get to biological tissue, it's not as predictable as gels, but we can still see general trends. So this is now the blue curve is our silicon sample, which is nice and well characterized. And then this is, the green is lung, liver, tumor, and then muscle. And you can see that that a lot of the trends still persist. Now, this is an area that we're still kind of pursuing because we have to apply a lot of different statistical methods to be able to pull out the actual values of this. So that this work is kind of still in progress. The other thing that we've noticed is that there's another mechanical property we can measure, and that is creep. Where creep is this factor that, for instance, if you have a viscoelastic deformable medium and you apply very quickly a force to it, like you try to squeeze it, or maybe it's a sponge, filled with water, and you try to squeeze it as quickly as you can, you know that right away you can squeeze it a great distance, but then it gets sort of harder and harder to squeeze out the last bit of water. And that's essentially what's happening here. If we turn on our field, we see that these particles will move, but then there's this element, this slope here, which is indicative of creep as that field is still applied, and then we switch it off. And so we're starting to also look at how we can measure creep of different tissues just with this technique. Okay, well, I always like to pull up this slide and call these blinking tumors. So there was an undergraduate in my group when we started this whole project, and he thought it was going to be the coolest thing, is that we can someday have images of blinking tumors where the nanoparticles would be localized to these tumors, and as we're switching the fields on and off, the areas of the tumor would essentially be flashing or blinking. We're not quite there yet, but we do notice that for different tissues, we're going to see these different rates of blinking and different signal intensities. I don't have, unfortunately, these used to be movies, but they're giving me problems. But we could see that these effects would be actually flashing on and off. I just wanted to put up kind of a segue into Professor Wei's talk. He's been a longtime collaborator of ours and has been working on different types of magnetic nanoparticles that could be used. These are magnetic gold nanostars. They're gold nanostars that have an iron oxide core. And these are interesting because perhaps potentially we can use these to look at mechanical properties in single cells. So we've been looking at developing techniques to be able to identify those nanoparticles. This is now optical coherence microscopy. And the gold nanoparticles actually show up quite well in 2-photon imaging. So we can identify where those particles are located within individual cells. And what we've been able to do then is build a small magnetic solenoid around our optical coherence microscope, and be able to switch that modulation on and off. And if we turn that modulation on, we see a very sharp spike at 90 hertz where we're modulating. And we can start to be able to identify which cells are labeled, and ultimately try to characterize their mechanical properties. Another area that's still ongoing in my group is that we can use the same type of magnetomotion as therapy. So perhaps people know that if you turn up the frequency or the field strength of this magnetic field, these iron oxide nanoparticles will start to modulate faster and faster. Well, at some point, the tissue isn't going to allow them to move. They're just trying to modulate so quickly. But the internal magnetization of the particles is still switching with the magnetic field. And that process causes the particles to heat up. So they can be used as hyperthermia agents as well under those conditions. Yeah.
Couple questions about that
last one?
Yeah.
Yeah.
So what's the idea of gold coating the iron oxide?
It really is to allow us to be able to localize where those particles are with some other way, some other modality. So the gold allows us to be able to identify, where those particles are with 2-photon. And then that ultimately allows us then to know, okay, that those particles are in and around our cell, and where we can then be able to sample those modulations.
I guess you could also say that there's a hope that, so far the Magnetomotive technology that Dr. Boppart has been showing is really based on the motion of the tissue. You're pushing the tissue up and down, but you might be able to see the individual particles with that same motion, so that greatly improves your spatial resolution.
If they're iron oxide low, I mean, you can see a single gold nanoparticles strong backscattering.
Sure.
So
that's right.
Yeah.
That's right. That's right. Yeah. So you're combining the best of both capabilities, both physical properties. Right. High scattering, and the magnetomotion.
Right. And what is the technique to localize these particles in the tumor? Like, what's the mechanism for it? Because you're getting a strong flaking signal, but the particles at the end of the day are going to be a way, I guess, of localizing the tumor, right? Finding the tumor in the blinking. So how do you inject them to the blood, and they select because of their size, they selectively go to the tumor?
Yeah, so we can target these just like we did with the bare iron oxide particles with different antibodies or other types of ligands, and those then can be localized. We can, but we haven't yet done types of intravital microscopy where you can look, at how these will localize, in a window model, for instance, in the tumor. I gave sort of the scenario that we can use the iron oxide properties to detect them with MRI on the whole body, and then use these types of techniques for the microscopic analysis as well. So one of I think the general principles when we design these agents is you always want to have some sort of a known validating method to know where they are, to know where how they're behaving. And in this case, we use 2-photon or an OCM to try to localize those. Oftentimes, you can do histology and with a Prussian blue type staining, which will stain the iron oxide a very specific purple color. So whenever you design your experiments, think about that. Think of having some validating way that everybody's going to know that works, and then test that against your new technique. So I think it's also kind of an interesting question, though, is we know that we can heat these up and they'll be hyperthermia agents. But what kind of modulation, what kind of change in function can we instill just by using the magnetomotion property itself? So we know we have nanometer scale, we can go up to micron scale displacement of particles. How might that change the function of the cell? And that's a kind of an active area for us to kind of think about still. We wanted to know where these particles go. So I just love electron microscopy because of the details. We wish we could have these in other modalities. But we set out to hunt for where these particles went. And so these are 20 nanometer iron oxide nanoparticles. These are macrophages that have engulfed those. And we started looking at higher and higher magnification of these really interesting cells. And the first thing you see is that cell membrane, is not as smooth as we think it is. There may be artifacts from the preparation here, but it's actually quite convoluted. So we think that we could identify very small, roughly 20 nanometer size particles. This is actually closer to about 200 nanometers. But this is our best guess at trying to find, particles, iron oxide particles, on the membrane of these cells, macrophages. We also did TEM, which gives us in cross-section, and we can clearly see the iron oxide on TEM. That's the dark signals. And these have been engulfed by the macrophages, but then here are some iron oxide particles on the membrane. So this is our best attempt to be able to find, we think these are the iron oxide particles on the membrane, and that's one, from SEM. Now, the other way, as we said, we wanted to be able to understand how these particles are interacting at the single cell level. We used some of Professor Popescu's technique to look at the phase of the light going through these cells. And so this is diffraction phase microscopy of a control macrophage. So a macrophage without any particles, and these are actually movies. You'll have to believe me, but that's playing right now. And you don't see any change. If we look at the macrophage that has particles, you can see this flickering. Okay, so you can see that there is movement of this cell that's detected with phase microscopy. And if we use the diffraction phase microscopy to kind of highlight that movement, you can see that there are areas where there's more movement. It all seems to be around the periphery of the cell, and maybe it corresponds to these iron oxide nanoparticles located on the membrane. So we then looked at trying to assess where the mechanical properties of the cell itself, and in this case, we used nano beads that were a couple hundred nanometers in size to give us a little bit larger particle, a stronger signal. And these beads, were localized in many different positions. This is a collection of beads here. This is the cell nucleus. So this is kind of the cell shape right here. And then we looked at, with a 5 hertz excitation frequency, we looked at the frequency spectrum. And at position 1, over the nanobeads, we saw very distinct signals at 5 hertz. And we saw, of course, harmonics as well. As we went further away from that position, that signal fell off. And over here in position 4, or even outside the cell, we didn't see any signal. So this was quite promising for us that we were localizing the signal within individual cells. And we sort of had could realize that there was some spatial modulation going on here. So it was essentially tenting. We were kind of pulling on these beads, and the area surrounding that bead, of course, was moving too, but not that far away. We saw very little signal. And what this led to, of course, is can we actually begin to identify a natural frequency of oscillation? So, as in this case, we were parked over that bead, but we were now varying the excitation frequency, and we saw that it also kind of peaked around 5 hertz. At about 1 hertz, about 10, 15, 20 or so, that amplitude of that signal was falling off. So the resonant frequency of that single cell was, to our estimate, around 5 hertz. And that's very different from the magnetomotive OCT measurements of tissue, which happen to be in a range of about 100 hertz. So the tissue as a bulk seems to be stiffer than that single cell that we were measuring. Okay. Well, let me just kind of go through this couple last examples where we took these nanoparticles and now we put them into microspheres. And we do this for a number of different reasons. These microspheres are bigger, so they're going to provide us a different type of contrast. We can see here the iron oxide in the core. It's an oil-filled core, and these are about 1 micron, 1 to 2 microns in size. And they're made by actually sonicating with an ultrasound horn between this protein and this oil interface. And it essentially makes a milkshake, makes these small microbubbles. This formation of this microemulsion cross-links these proteins to form the shell. But it gives us a lot of flexibility in terms of what we can add to the shell or put inside the core. So we can do things like make the shells out of different materials. We can have different inner cores, mostly oil, and we can modify the surface with PEG, with folate, with antibodies, many different types of techniques to design these contrast agents. And these are just some examples. We've been trying to design better agents for OCT, and so of course we add scattering particles, carbon, melanin, gold, here's the iron oxide, in an effort to increase the optical scattering properties of these microspheres. And these are the SEMs and the TEMs of those individual microspheres. Now we did a couple of early studies with these microspheres where we injected them into the bloodstream, and if we were imaging the liver, we saw that with the agents, all of a sudden the macrophages in the liver were taking these microspheres out of circulation, and so the liver scattering properties increased. And what we believe are seen here are really the sinusoids in the liver, that have the blood flow through them and are not taking up the microspheres. If we look at, it's kind of hard to see here, but we're looking at a pair of blood vessels in the colon of this mouse, or the intestinal wall, and with the injection of the microspheres, we see this increased optical scattering as the microspheres are circulating. And just outside the blood vessels, we don't see any change in scattering. It's what we would expect, because these microspheres don't get out of the bloodstream. They're too large. Usually the particles have to be less than a few hundred nanometers to actually get out of the bloodstream. We do know that these get taken up by macrophages. So the macrophages that have been exposed to these microspheres, you can see have all these microspheres engulfed, and they're breaking them down in that process. So the liver is filled with macrophages, and that's why a lot of these microspheres end up in the liver too. Now these magnetomotive microspheres, the ones that have iron oxide, do give us this magnetomotive signal. And these are the signal coming from what we believe individual microspheres that are now modulating within the tissue. And just like we did for the nanoparticles, we can conjugate, in this case, RGD, the RGD peptide, to the surface of these microspheres. And we can determine, we figured out what is the best polypeptide chain to be able to target tumor cells with this RGD. Now this is HT29 tumor cells. These microspheres had a dye in them, so we just did fluorescence microscopy, and we could see that with this RGD and then the 6 polypeptide chain was the best for targeting these cells. We can then take these microspheres. They show up, again, as discrete elements, as either within a gel. This is taken up by macrophages, and this is actually in vivo intratumor injection. So again, seeing very discrete kind of punctate type signal. And a lot of our effort in these microspheres is to recognize that they're not going to get out of the bloodstream, but with RGD they can target angiogenesis in cancer, and those new blood vessels are going to overexpress RGD, or the alpha V beta 3 integrin, which is where RGD is targeted. The same is true in atherosclerosis. So this type of lesions, these fatty streaks and atherosclerotic lesions, are going to overexpress these integrin receptors, which we can target. So we did an animal study that induced atherosclerosis. We took, this was a control rat, fed a normal diet. We took out the aorta, and we washed these targeted microspheres over it, and we saw very little, maybe just a few stuck on the outside where there's fat. That's in contrast to a rat that was fed a US diet, high fat, high cholesterol, and we were able to see very kind of punctate areas that corresponded to early plaques. So this is the histology that's kind of broken, but this was an area of plaque and an area that was targeted with these microspheres. So we're thinking this could also be used then to target atherosclerosis, even early changes. And because the microspheres are quite large, they can carry a drug payload, and we could potentially rupture those to deliver sort of anti-inflammatory drugs or other cytokines locally to the site. Now, just like the nanoparticles, these microspheres show that negative T2 contrast, and because they have a lot of iron oxide, they actually make organs disappear. And it's really amazing. We didn't believe it at first. This is a rat, and what you're looking at is sort of the liver, both in coronal and cross-section. And this is without agents. When we add the microspheres, we know they're all going to the liver. And so they cause such a strong negative T2 contrast that the liver sort of just disappears. And this is the same section of the rat, but the liver is gone because of that contrast. This is individual nanoparticles that collect in the kidneys. And so without the agents, we can see the cortex of the kidney. And then with the agents, we see this darkening of that area. Okay, and finally, these particles, these microspheres, also function as ultrasound agents. They look a lot like the microbubbles used in ultrasound. And so we used a high-frequency ultrasound system that with standard gas-filled microbubbles, you can see the microbubbles show up in the vasculature. And it's hard to see here, but from your notes, you should be able to see that as the microbubbles enter the circulation, the scattering goes up, and you apply these high-intensity ultrasound pulses that will rupture the bubbles. And the scattering from the bubbles drops right after that. So that's how you know you rupture these bubbles. We could see the same effect with our oil-filled microspheres. We see this green signal everywhere is where the bubbles are present. And this was a surprise to us because microbubbles under ultrasound will sort of oscillate back and forth. But if you fill them with oil, they're not going to do that very well. And so we weren't sure we were going to see such a signal. But indeed, we saw that the signal went up as we injected our oil-filled microspheres. It took, in fact, about 20 rupture pulses instead of just a few. But we finally were able to rupture a portion of those with ultrasound. And if we took out this tumor, we could then see the magnetomotive signal present. We did fluorescence microscopy, and we could see both a mixture of some intact microspheres, and then we saw this diffuse haze of fluorescence, which was the oil core, the Nile red that was in the oil core that was now diffusing out because the microspheres were ruptured. So these agents, these microsphere agents, are actually quite multimodal. All right. Well, the last part, how we can detect these agents, is coming back to our spectroscopic OCT that we talked about last week. And the point was that the envelope gives us something about the structure of the tissue. The carrier wave can tell us about the wavelength content. And this is just one experimental demonstration where we had a cuvette filled with a near IR dye. And you can see that the laser spectrum shown in green compared to the laser spectrum that is transmitted through the dye. And you see that there's a loss, there's an absorption of wavelengths at the shorter end of the laser spectrum. And so that causes our centroid shift to go from here to here because of the absorption of that dye. Well, that can be used in a quantitative manner. And again, if we have our dye, our molecular dye, that essentially splits the laser spectrum, we can look for that shift quite distinctly. And of course, that shift is going to occur somewhat linearly as we change the dye concentration. All right, well, that was used then with spectroscopic OCT to identify the presence of a dye. We did the typical high school experiment where we took the celery stalk and we we cut it off, we put it in the beaker filled with dye. And because of the natural transpiration of the plant, it took the dye up into its vascular system. And so what you're looking at is a cross-section of a celery stalk. This is in bright field. And the dye, because it's a fluorescent dye, we could actually see the dye is being taken up through the vascular system. And we can see that spectroscopic OCT can identify the presence of the dye within that same vascular system. Now, the same technique can be applied coming back to these nanoparticles, these gold nanoparticles. And as I think Professor Wei is going to talk about his work in gold nanorods that have very distinct absorption spectra that correspond to either a longitudinal plasmon resonance or a transverse. And this, in fact, can be tuned by changing the size of the nanorods. And we can have a very specific and narrow spectrum. And that's what we want to use to detect. And we've done a number of studies over the years where these nanorods can then be detected spectroscopically based on this spectral shift in the OCT signal that we recover. And this is just one example of increasing concentration of nanorods. Finally, we're not limited to nanorods. We did one study looking at single-walled carbon nanotubes that have a very strong absorption as well. And we can actually take these nanotubes, add a magnetic domain, control it with a magnetic field, and again, try to develop these types of multimodal contrast agents that can be detected with MRI, can be detected optically, or potentially with OCT as well. So just to summarize, this type of optical scattering-based images in OCT are frequently plagued by this low contrast. And so we can develop and design novel types of agents that allow us to alter the scattering properties, the absorption properties, or modulating properties, and improve this sort of diagnostic ability of OCT. There's also the significant therapeutic applications with heating up tissue or delivering drugs. Now, I talked about exogenous contrast agents today. There's really going to be also a whole nother realm of endogenous spectroscopic detection with spectroscopic OCT or these nonlinear techniques like CARS or second harmonic generation as well. So last week I covered this again. Today I wrapped up everything, and hopefully this gives you a good picture of the technique and how we can really improve its imaging capabilities with OCT. And again, just to thank my group, our funding sources, our collaborators. We really need the help of chemists when it comes into designing these types of agents, and appreciate our collaborations over the years. So thanks again for your attention, and any questions I'm happy to answer. Yeah.
How would you use nanoparticles?
That's a good question. So as much of the work as we spend trying to figure out, can they improve contrast, where do they go, an equal amount of work has to go into sort of its clearance, the biodistribution and clearance. And that really is an area that I think is still under-addressed. We know, to the best of our ability, they can target to tumors. We know that they are cleared through the liver or through the kidneys, but a lot of studies need to be done in terms of just how fast does that happen, what's the long-term potential side effects of these agents. A lot of that has been done for our FDA-approved agents, and as far as we know, they're safe at those doses. We are constantly trying to figure out, can we use those very small doses to provide the best signal. So this is an area I think that many of you perhaps could help solve, is that we need to get a better handle on designing the agent that's going to come in, target, provide that signal, and then get out, or get broken down into sort of natural byproducts. For the most part, the liver and the kidneys do their things well. They'll take out the macrophages in the liver will take out any of these particles. They start to break them down. They're excreted then. Long term, we know that a lot of these particles aren't always retained, but sometimes they can be. They can accumulate over time. The kidneys, have a filtration system, so large particles can actually occlude the kidneys. Smaller particles will be excreted, just through the kidneys, filtered out into the bladder, out of the urine. But again, it's going to change, and it changes in disease. So we can know all this in a healthy animal or a healthy human, But anytime you have someone with disease, which is what you're trying to image in the first place, that's going to be a very different picture. So we don't know enough about cancer in general, let alone to how it changes our physiological systems in individual patients. So you could have one cancer patient where it may excrete easily, the next patient it doesn't. We just don't know enough yet. So that's a tough problem. Any other questions? Yes. Yes.
So if there's different size or geometry particles, would that affect the resonance that you observe?
That's a good question. So from what we could tell, the size of the particle will allow us to exert a stronger force on that particle itself. The particles themselves are quite small and sort of massless compared to the tissue that they're moving. So they don't really alter that resonant frequency significantly. But it does allow us to kind of maybe move that tissue further, a further displacement. But that is a really interesting area because the tissue is so heterogeneous that right now we're trying to figure out, is there a way, can we probe even on the micron scale and get one measurement of viscoelasticity? But that may be very different from the tissue just a millimeter away. And so how do we deal with sort of that heterogeneity as well?
Theoretically, you could have different sizes to target different molecules. That way, one injection target almost anything that you're looking.
Yeah. Although I think, my guess would be the heterogeneity of the tissue is going to be greater than the differences in signal that you would see from different size particles. So it's one of these things we still have to think about. How do we, are we measuring the micron scale or are we measuring the bulk? It's all one connected system. So even though we're perturbing in one area, some of our measurements sort of reflect still the bulk tissue. And that's just because it's all coupled. Okay.